Morning. In this video, we are going to explain a second type of plasma membrane receptors, which are the tyrosine kinases or receptor tyrosine kinases. To start with, we have to remind you or tell you, if you don't know, that the kinases are a group of enzymes in the human body. And just like any enzyme, they catalyze a certain chemical reaction. The chemical reaction they catalyze is a chemical reaction called phosphorylation of their substrate. You know that every enzyme has a certain substrate, a specific substrate on which it acts. And we will be discussing this in more details in Unit 3. And their main function is to phosphorylate the substrate, meaning to add a phosphate group to the substrate. But this phosphate group, so phosphorylation means the addition of a phosphate group, But this phosphate group should be taken from hydrolysis of ATP, meaning that this phosphate might be of organic origin because ATP is an organic molecule. There are other enzymes that cause phosphorylation, but these enzymes are called phosphorylases, not kinases. And these enzymes also add phosphate to the substrate, but they usually add inorganic phosphate, meaning that it's not derived from an organic molecule. As we said in the previous video, phosphorylation is very important, whether it is phosphorylation through enzymes known as kinases or through the enzymes that are known as phosphorylases. The addition of phosphate to a substrate or phosphorylation is important because it might either activate the substrate or inactivate the substrate. So phosphorylation is not always about activation of the substrate, but it might in some instances lead to inactivation of a substrate. And by the way, that substrate may be another enzyme. We will see all these details over the course. The kinases that are present in the cell membrane are considered plasma membrane receptors. So they lie in the plasma membrane and they act as receptors. And we will see how these enzymes act as receptors. So some of the receptors of the cell membrane actually act as enzymes. We will see this in detail on the next slide. And these receptor tyrosine kinases, like many of the protein receptors in the cell membrane, span out the whole thickness of the cell membrane. But does the receptor as a whole act as enzyme? No, it's only the cytoplasmic part of the receptor that functions as a tyrosine kinase or as an enzyme that catalyzes the transfer of phosphates from ATP. But from ATP to which substrate? To a specific substrate, because you know that every enzyme has its own substrate, and the specific substrate of the receptor tyrosine kinases, of course, is the amino acid tyrosine. Now, of course, just like any receptor, this receptor has a specific domain where the ligand or the signaling molecule will bind. And we will know what the ligand is. But before the ligand binds to the domain, of course, when the ligand binds, it binds to the specific shape on the receptor molecule that fits with the ligand or the signaling molecule. So before it binds to the specific domain on the receptor, the receptor exists as two individual units, meaning that it's not one block, it's not one entity, it is divided into two individual units. But after the specific ligand binds to the specific domain, and notice that I'm using the adjective specific. So once the specific ligand binds to the specific domain, these individual units will aggregate together, will join together. When we say aggregate together, we mean will join together, and they will form a dimer. And a dimer is a bigger molecule that is made of two halves. And when the two halves of the receptor bind to each other, this is when the enzymatic cytoplasmic part of the receptor will be activated. This is when the tyrosine kinase region will be activated. So the binding of the ligand to the receptor aggregates the two dimers and activates the cytoplasmic portion of the receptor, which is the tyrosine kinase. And the tyrosine kinase, when activated, will do what? The tyrosine kinase will bond to ATP. And after having bonded to ATP, the sequential steps are activation of specific proteins, one after the other, like a domino effect, and that's why we say that they are relay proteins. So when the tyrosine kinase is activated, it binds to ATP. When it binds to ATP, it takes the phosphate group and transfers it to the substrate, which is a protein. And we call this a relay protein because this relay protein will itself activate a following protein and so on until we obtain the full cellular response. And this is an example of course, signal transduction pathway. So this is the end of the signal transduction pathway. When you obtain a response, this is the last step of the signal transduction pathway or the cascade. Now, these are pictures showing the receptor tyrosine kinases. So here, these are the two halves of the tyrosine kinase enzyme. This is the ligand or the signaling molecule. And these are the binding sites or the domains where the specific domains where the specific ligand, you can say here instead of signaling molecule, you can say the specific ligand. And then we have an extracellular part, which is the domain, a transmembrane part, which is an alpha helix, and a cytoplasmic part. And the cytoplasmic part of the receptor is actually the enzyme or the tyrosine kinase. And the tyrosine kinase has several tyrosine molecules or tyrosine, we call them tyrosine residues. And these are inactive up till now. But the second step is that the signaling in number 2 here, the signaling molecule, as you see here, the signaling molecules two signaling molecules actually will bind to two receptors, extracellular receptors, and they will fit in. When they fit in, the two halves of the tyrosine kinase join to each other and a dimer is formed. When the dimer is formed, this is an activated tyrosine kinase enzyme. So what happens is that there is binding with ATP and conversion of ATP into ATP. 6 molecules of ATP are converted into 6 molecules of ATP, and therefore there will be 6 molecules of phosphate that are released. But why didn't we write them like this? Because they are not released. They are actually bonded to the tyrosine immediately. So the 6 phosphate groups that result from hydrolysis of 6 ATP molecules will bind to the 6 tyrosine residues, and now we have the fully activated receptor. So the first step in activation of the receptor is the joining of the two halves together and the formation of the dimer. The second step is the binding with ATP and hydrolysis of ATP into ADP. And the third step for full activation is the phosphorylation of tyrosine. So here we have 3 steps of activation of the enzyme. First step in the activation of the enzyme is the formation of the dimer. The second step is binding to ATP. And the third step is to phosphorylate the tyrosine residues. And after the tyrosine residues have been phosphorylated, as you see in picture number 4, the proteins, the relay proteins that will induce different cellular responses will be activated by being phosphorylated. So you can see at the bottom of the picture these 2 proteins that are inactive, and they bind with the phosphate on the activated receptor, and they get activated by being phosphorylated. Actually, the whole process of activation here is through phosphorylation. Here we have 2 different relay proteins. One of them, as you see, initiates cellular response 1, and the other initiates cellular response 2. And this cellular response is nothing but a sequence of proteins that will be phosphorylated, resulting in a sequence of chemical reactions until we obtain a product that provides the cell with the ultimate cellular response. So another characteristic of the receptor tyrosine kinases is that you can obtain multiple responses. It's not only one relay protein that can act or that can bind with the activated receptor, but several proteins, each of them being capable of activating a cellular response. But sometimes the enzyme is not a hydrophilic hormone, or the ligand molecule is not a hydrophilic hormone. But it is a hydrophobic hormone, like the steroid hormones, for example. Like the sex hormones and cortisone. Steroid hormones, of course, we have the sex hormones, whether they are male hormones or female hormones, and we have the cortisol group. These are steroid hormones, and they have their own receptors to initiate their own chemical pathways. But because these steroid hormones, whether the sex hormones or cortisol, because they are steroid, so they are hydrophobic hormones. And since they are hydrophobic hormones, then they can move easily through the hydrophobic interior of the cell. But in order to do this easily, not only do they need to be nonpolar and hydrophobic, but they also need to be small in size. So the first characteristic of these hormones, which are actually considered the ligands because they are the signaling molecules that will bind with and signal or stimulate a cellular response. They are small molecules, they are hydrophobic or nonpolar, and they are considered, of course, ligands or signaling molecules. Of course, they can diffuse directly through the hydrophobic interior of the plasma membrane. And in such a case, they do not need to have receptors on top of the cell, on the cell membrane, but their receptors are intracellular receptors. Examples of such hydrophobic chemical messengers include, as we said before, the steroid hormones, sex hormones, and cortisol, as well as an important chemical that is produced in the body, which is NO, or nitric oxide. N refers to nitrogen and O refers to oxygen. And this is a gas produced in the body This is a gas produced in the body to dilate the blood capillaries. It is important because it dilates the blood capillaries, thus increasing the blood flow. When the ligand binds with the receptor, this happens inside the cytosol, as we said before, because we will be seeing on the next slide how the receptors are intracytosolic receptors. They are not on the cell membrane, but they are inside the cell. And this ligand receptor complex will not only be inside the cytosol, but it will pass through the nuclear pore, meaning that the complex itself, the ligand, which is a small molecule, and the receptor, which is also a small molecule. Why are they supposed to be small? First, the ligand is small to pass through the cell membrane easily, and the receptor is small to be able to pass through the nuclear pores. The nuclear pores are the pores present in the nuclear membrane. Having passed through the nuclear pore, it will find itself inside the nucleus, and it will bind as a whole, as a complex, to chromatin. And chromatin, as you know, is DNA plus nuclear proteins. And this will result in turning a gene on or turning a gene off. Turning a gene off or turning a gene on, this is what we call regulation of gene expression. So these complexes are capable of, not only are capable of, but act through turning genes on or turning genes off. And therefore, this is the mechanism of action of steroid hormones. Steroid hormones do not act like peptide hormones by formation of cyclic AMP and inducing biochemical reactions in the cytosol to produce a final cellular response, but they act through turning on a gene or turning off a gene, and therefore through activation or inactivation of all the processes afterwards, which are transcription of messenger RNA and then translation into a peptide. Of course, we know that one of the major examples is the estrogen hormone, just because the estrogen hormone is a lipid hormone, and therefore it is it is a steroid hormone, and therefore it is a lipid hormone. It is a non-polar hormone, and it is a hydrophobic hormone. Similarly, the male hormones, which are the androgens. But there are two other substances that act the same way as the steroid hormones, although they are not steroid hormones. The first one is very important and is very dangerous, which is bisphenol A. Bisphenol A is a substance that is present in many, many plastic substances, like plastic bottles and plastic boxes, for example. And it mimics estrogen hormone. The seriousness of having bisphenol A inside the human body is that it mimics chemically, of course, the estrogen hormones, and therefore it can bind with the estrogen receptors and set up an estrogen signal transduction pathway, meaning that it can act exactly like estrogen, bind with the receptor, pass through the nuclear pore, bind with a certain another receptor on chromatin, turn on or turn off a certain gene. And therefore, if, for example, a pregnant female is exposed to high levels of bisphenol A, this might result in problems in fetal development in human beings, like feminization, for example, of a human fetus to a certain extent. Feminization, because it acts like estrogen. How can a pregnant woman or an individual in general have bisphenol A in its blood? This is through, for example, having bottles of water that are forgotten in the cars in hot weather, exposed to sun rays, and then we cool them down again and we drink from them. Or, for example, if you use plastic boxes or plastic baby bottles, milk bottles, you put them in the microwave and they are not essentially microwavable, meaning that they will be degraded and broken down partially, of course, by even if they are not distorted in shape, some bisphenol A molecules will be produced from the high energy of microwave or from the high solar energy and will dissolve in water. The third example is the thyroxine. Thyroxine hormone acts like steroid hormones, although it's not a steroid hormone. It is a peptide hormone, but it has a mechanism of action that is similar to that of the steroid hormone. So there is uptake of thyroxine. First, there is an uptake of thyroxine by a carrier that binds a carrier on the cell membrane, a protein on the cell membrane that uptakes thyroxine. And that's why we call this a carrier-mediated uptake, meaning that this specific carrier protein will bind specifically with thyroxine. And this carrier protein is on the cell membrane, but this carrier protein itself is not a receptor. Why do we say this? Because it's not capable of inducing a cellular response. So it is just a carrier molecule for thyroxine to help thyroxine, which is a hydrophilic molecule, to be carried inside the cell. And that's why we call this uptake a carrier-mediated uptake. So the function of this cell membrane protein is just to uptake thyroxine. But why do we need all this complexity? Because thyroxine is a hydrophilic peptide hormone, and therefore it cannot enter easily inside the cell membrane. But once thyroxine is inside the cytosol, it will act like steroid hormones. This process of carrier-mediated uptake of thyroxine is energy dependent and sodium dependent. So the transporter is dependent on the presence of sodium ions and the simultaneous transport of sodium ions in the presence of energy. We can see details of this later on, of several processes of primary and secondary active transport where we need sodium ion to be transported at the same time. Here, for example, we can see how a steroid hormone can enter easily. A steroid hormone, this molecule can enter easily through the cell membrane and then bind with a receptor inside the cell. This receptor is an intracytosolic receptor, and you can see how at the beginning this receptor was empty and then it was bonded to the hormone. And then together, although this is a complex, yet it is small enough to pass through the nuclear pore and then bind with the chromatin inside the nucleus and therefore affect DNA transcription. This diagram shows the same, but here we have a lipid-soluble molecule coming from the blood. For example, a hormone coming from the suprarenal gland and being transported into the blood. So the first step is that it diffuses through the plasma membrane, helped by the fact that it is hydrophobic, and then it binds with a cytoplasmic receptor and forms a complex. Then it enters the nucleus through the nucleus pore and binds with chromatin and affects gene transcription, and the transcribed messenger RNA will result in the formation of a particular protein. We've talked about transduction before, and we said that once the activated receptor converts the molecular reception of the signal into the cellular response, this is called a transduction. So transduction is the process where deactivated receptor, meaning the receptor that has been bonded to the signaling molecule, will convert that binding or that process of reception, just reception of the signaling molecule, into a cellular response. So transduction here means conversion or change. This transduction sometimes occurs as a single step, but most often it requires, as we said before, a sequence of biochemical reactions in which the product of every chemical reaction will be the substrate of the next chemical reaction. So a signal transduction pathway. So a signal transduction pathway is a complex process. Yes, it is a multi-step process, but there is, as we said before, not only is it a complex process, nor only is it a multi-step process, but it's also a process where amplification occurs, where we obtain at the end of the day a major effect. And why are we saying this? Because we start by a small number of ligand molecules or extracellular signaling molecules, and we end up by having thousands of product molecules. So there is a cascade effect or a dominoes effect. And we said before that whenever we are asked about the cascade effect and the dominoes effect, we have to talk about two things. We have to talk about the sequential biochemical reactions, meaning that just like every domino pieces pushes the next one to fall down, every chemical reaction will activate another chemical reaction. And this is actually the dominoes effect. But it also includes the sense of amplification. So cascade or dominoes includes the sequential effect, and at the same time the amplification effect. So the signal is being amplified. This process is usually present in higher organisms, meaning that the cellular transduction pathway is mostly present in higher plant and animal cells and in yeasts, but in bacteria, it's not as well developed as in multicellular complex organisms. There is, as we said before, they are very interested in AP biology, in this process of evolution and to prove that everything evolved from a common origin. And they always look for similarities in structure or similarities in function or similarities in, here, for example, biochemical pathways. And therefore, you might be asked as usual about the evolutionary, this is a common question, and unluckily you're always confused about the answer, the evolutionary significance of the biochemical pathway. So this is an important question, very common question, although the answer is very easy. The evolutionary significance of the biochemical pathways comes from the fact that there is great similarity between the biochemical pathways and that they are present in a diverse number of organisms. So it's widespread in living organisms, and meanwhile there is similarity. So we have to focus on two main points. The first one is the widespreadness of those chemical pathways, and the second point is the similarity of those biochemical pathways. This is very interesting to prove that there was some common ancestors in the past. And now we are going to study an example of a signal transduction pathway where a G protein coupled receptor is included, and this is the chemical pathway of glycogenolysis in hepatic cells. Of course, hepatic cells, as you know, are liver cells, and glycogenolysis, lysis means breakdown. This means that liver cells will hydrolyze glycogen into glucose, and this happens when the blood vessels do not contain enough glucose, and that this glucose will be transported into the circulation. So this does not occur on liver cells, do not do this on their own, but they do this under the effect of epinephrine hormone. So epinephrine is the hormone that will stimulate the signal transduction pathway that will result in glycogenolysis. So again, glycogenolysis, as you see on the slide, is the breakdown of glycogen into simpler units or simpler monomers, which is glucose, and it is released by the liver cells into the bloodstream whenever the body needs of glucose increase. This is stimulated by epinephrine. So epinephrine will bind to a receptor that will activate a G protein. So we can say that epinephrine is the specific ligand molecule. Again, specific ligand molecule, or that it is the signaling molecule. And since the receptor to which epinephrine binds will activate the G protein, then this receptor is a G protein coupled receptor. And since it is a G protein coupled receptor, so it will achieve the cellular response through the formation of cyclic AMP that is considered the intracellular secondary messenger, because the primary messenger in such a case is epinephrine. Cyclic AMP will amplify the signal through the cell. It will distribute the signal, meaning that, and from here comes the process of amplification. If you want to understand how amplification occurs, we started by one epinephrine molecule, but after the G protein has been activated and cyclic AMP has been produced, this cyclic AMP will bind with several substrate molecules and will amplify the signal throughout the cell. And therefore, there is a cascade of chemical reactions that occur afterwards to produce thousands of molecules of glucose. So here we have the first step is binding of epinephrine. This is one molecule, and then the process of transduction. Inactive G protein is converted into active G protein. And this process of activation does not occur in only one G protein portion, but it occurs in 100 molecules. So you will see as we go forward that the number of molecules will increase. And then this active G protein will convert inactive adenylate cyclase enzyme into active adenylate cyclase enzyme. Again, we just have 100 molecules. And then this active adenylate cyclase enzyme will convert ATP into cyclic AMP. But the 100 molecules of adenylate cyclase, the 100 molecules will not only activate 100 molecules of ATP, but they will activate 10,000 molecules, meaning that every enzyme, adenylate cyclase enzyme, every molecule of the enzyme can activate 100 molecules of or convert 100 molecules of ATP into cyclic AMP. And this is normal because, as you know, the enzyme does not stay permanently on this, or the substrate does not stay permanently on the substrate. And every now and then, once the product is formed, a new substrate molecule is formed. So how can this be explained, this step? This means that every enzyme molecule can change 100 molecules of ATP into 100 molecules of cyclic AMP. So if you start by 100 molecules of enzymes, then you will end up by having 10,000 molecules of ATP, and then ATP of cyclic AMP, and cyclic AMP will phosphorylate inactive protein kinase to convert it into active protein kinase, and then active protein kinase This is inactive protein kinase A that has been phosphorylated into active protein kinase, and then active protein kinase will activate inactive phosphorylase. And here is the phosphorylase I have talked to you about. And that phosphorylase enzyme is an enzyme that can phosphorylate, but using an inorganic phosphate. And then at the end of the day, we have glycogen phosphorylase enzyme that is activated, that phosphorylates glycogen to convert it into glucose. You will never be asked to write this or to learn this by heart or to memorize the steps, but it's only about understanding the fact. What concepts can we deduce from this diagram? We can deduce that signal transduction pathway occurs in 3 steps: reception, transduction, and response. That transduction occurs in a series of biochemical pathways, and this is what we call the cascade or dominoes effect, and that it all starts by reception or binding of a signaling molecule to a receptor, and at the end of the day, there is amplification or augmentation of, we start by one signaling molecule and we end up by having thousands of product molecules. And this is what we call the cellular response. So ultimately, the signal transduction pathway leads to a multitude of responses. And these responses might be products obtained in the cytosol, or as you saw in case of the steroid hormones, products obtained in the nucleus. Many signaling pathways will regulate protein synthesis by turning genes on or turning genes off, even if they are not steroid hormones. And we will see examples of this later on. What are the characteristics of a signal transduction pathway? This is very important. First of all, as we saw before, number 1, there is a signal or a signaling molecule or a ligand. So there is a signaling molecule, and then there is the conversion of the signal into a cellular response. The conversion of the signal into a cellular response is a series of biochemical steps that is called transduction. Of course, another important characteristic of the signal transduction pathway is that it is highly specific and highly regulated. Highly specific and highly regulated. Highly specific because it starts by the binding of a specific ligand with a specific receptor. Highly regulated because it's a series of specific biochemical reactions that occur in a specific order and that are catalyzed by a specific enzyme. So you have a series of specific chemical reactions, specific chemical reactions that have a specific order or a specific sequence and that are catalyzed by specific enzymes. So it is highly regulated. And then, of course, one important characteristic of the cellular responses is the cascade effect, releasing thousands of product molecules. And as we said before, the cellular response might be an alteration in gene expression on or off. Any kind of cellular activity, like increasing a certain protein activity or just increasing the synthesis of a particular protein. And we've said that they have an important evolutionary significance. All this is nothing but, or the content of that slide as a whole is a wrap-up of what we have already said. And then we are going to start a new concept, which is the concept of transport across the cell membrane. So the transport is the movement of substances inwards or outwards into the cell or outside the cell across the cell membrane. Although it might look simple, but we will study a lot about transport across the cell membrane. First of all, we have to know that transport is either a passive process or an active process. Passive process meaning that this process does not require any kind of energy, but an active process requires chemical energy that is derived from hydrolysis or breakdown of ATP. So passive transport is the first and very common method of transport across cell membranes, and it is the downwards movement of molecules from a region of a higher concentration to a region of lower concentration until equilibrium is reached. So if this is the cell membrane, for example, and if we have here 5 molecules of substance A and here only 1 molecule of substance A, and if substance A is capable of passing easily through the cell membrane, this is an important condition, because if it's not capable of passing easily through the cell membrane, then we will require energy to allow it to pass. We suppose that this substance A, for example, is a non-polar substance, and that it is a small particle. So it will pass from the region where it has a higher concentration to a region where it is in, or it has a lower concentration. And this sounds normal. This is what we call downhill, or this is what we call down concentration gradient. Concentration gradient means difference in concentration. So outside we have a high concentration, inside we have a low concentration. So this is the difference in concentration between 5 and 1, for example. So it is going down concentration gradient, or with concentration gradient. Gradient here means difference. There are several examples of passive transport that we will be discussing. The first one is simple diffusion. And in simple diffusion, the substance passes just like I have just explained, and it needs no protein channels or carriers. We have seen the channels for ions, we have seen the carriers for large polar molecules, but since the particle is neither an ion nor a large molecule, then it will be carried by simple diffusion. For example, oxygen. Oxygen is a small non-polar molecule. Carbon dioxide, it is a small non-polar molecule. So both gases, and this is important for the body because both oxygen and carbon dioxide are very important for the human body. They can just pass by simple diffusion. But sometimes when the molecule is large and polar, or the particle is an ion, as we have explained before, we require a carrier or a channel. So in case of large polar molecules like glucose and amino acids, then we need protein carriers. And in case of ions, we need channels. If you use a carrier or if you use a channel, then this process is no more called simple diffusion, but it is called facilitated diffusion. Again, facilitated diffusion, although it needs a protein carrier or a protein channel, yet it is passive diffusion. Why is it passive diffusion? Because it requires no energy, it requires no carriers, and it requires no energy only, although it requires carriers and channels. So let's say it again. So this, what I've just written is about simple diffusion. So simple diffusion, no energy, no carriers, and no channels. But in case of facilitated diffusion, no energy, but we have carriers and we have channels. And then the third type of passive transport is countercurrent exchange that we'll be explaining later on. Another type of passive transport other than diffusion is osmosis. And osmosis is a specific type of diffusion because it involves water only. So it requires no energy, it requires no carriers, it requires no channels, but it is specific to water. Thank you, and we will be continuing transport, the different types of transport on the following video.